Hello, my name is Nicholas Hartel, and I'm a PhD candidate at the University of Southern California. And my work is on protein methylation. And specifically, I'll be talking about how to discriminate two forms of arginine dimethylation that can be tricky to tell apart. So we know the central dogma of biology. We have our DNA to RNA to protein. And those proteins can get modified and do some function, such as metabolism. My work focuses on this part here, the functional protein or the post-translational control, specifically protein modifications. So what is protein methylation? Protein methylation can occur on two amino acids, lysine and arginine. For lysine, we have the monomethyl, the dimethyl, and trimethyl with the subsequent mass shifts down here. For arginine, there's also three forms. We have the monomethyl, then we have an asymmetric dimethyl with two methyl groups on the same nitrogen, and then the symmetric dimethyl with both methyl groups on opposing nitrogens. And you can see that the asymmetric and symmetric have the same mass shifts. So why is protein methylation an understudied post-translational modification? Well, the methyl group is quite small. It doesn't alter the charge. As you can see here, the addition of the methyl group, the arginine still has its positive charge. And because of that, it's not very bulky, so it doesn't add much mass to the protein. It's also a relatively rare modification. So because of that, there's low stoichiometry. So it needs to be enriched from a complex cell lysate. And as I just mentioned, it's trickier because the asymmetric and symmetric forms are isomers and therefore have identical mass shifts. And so they can't be told apart by mass alone. And protein methylation has gained a lot of interest recently because of what it's been found to regulate. So a really big one is RNA splicing. We see many RNA proteins involved in RNA splicing are heavily methylated. Metabolism, proliferation, even life and death for some cancer cells has been shown to depend on protein methylation. And so protein methylation is catalyzed by PRMTs, which are protein arginine methyltransferases. And there's 9 of those that are known. And so all of them can catalyze the monomethyl form of arginine, MMA. And then they can be divided into two groups, the type 1 PRMTs, PRMTs 1, 2, 3, 4, 6, 8. They can catalyze the asymmetric form, and the other PRMTs are called type 2 PRMTs, and they catalyze the symmetric form, such as 5, 7, and 9. And type 2 PRMTs can still catalyze the monomethyl form. And so this is a workflow for methylproteomics that we've developed. So we can start with some biological perturbation, such as knocking down a protein arginine methyltransferase, for example. Then we're gonna need to enrich these methylpeptides because they're low abundance. Then we run those samples on our LC-MS system. And hopefully we would search, find a bunch of methyl proteins, and able to quantify the perturbation that we did. So the problem comes with how to tell these two apart. As you can see, identical mass shifts, identical masses. It appears that there would be no way, but we fragment our peptides in a collision chamber before we measure the ions. And then based off those ions, we can reconstruct the peptide that was fragmented. That occurs in this collision cell over here called the HCD cell, or higher collision dissociation. And if you do that, you can get a neutral loss to occur, such as this. So by hitting these peptides with some high energy nitrogen gas in our collision chamber, you can get this moiety of dimethylamine to be kicked off. if it's ADMA. And for SDMA, you get a monomethylamine kicked off because the methyl group is on, there's only one methyl group on each nitrogen. And then even though these neutral losses have no charge and therefore can't be measured, the remaining ion has now a mass deficit that can be identified and then assigned as either ADMA or SDMA. So let's look at an example. So here's an example spectra from a dimethyl peptide we saw. Here's the sequence of the peptide up here. And we see all these Y ions. And then if you zoom in a little on the spectra, these inset tables show other ions that seem to be the ions that lost their neutral loss. And so they're denoted by a star symbol here that you can see. And if you take the difference of these ions with their respective parent Y ion, you'll see that for all of these, we got a difference of about 45.058, which corresponds to the dimethylamine neutral loss. So now we know that this dimethyl peptide can be assigned as an ADMA peptide. And for further evidence, we even see the isotopic envelope of the neutral loss ion here, which is the extra neutrons in isotopes, the natural isotopes. And this is just a characteristic of peptide. It's just further evidence that it's a real ion. So we wanted to validate this approach first with synthetic data. So we started with about 140 synthetic ADMA peptides, singly dimethylated, and also about the same number of synthetic SDMA peptides, the symmetric form. And when we looked for neutral loss, we got pretty good assignment with very little error rate. And in a previous data set, we mined the data to look at if we could see neutral losses in our own real data. And from these few enrichments we did get some pretty good annotation of ADMA and SDMA. In the strong cation exchange enrichment and the immunoaffinity purifications. So what can we take away from this so far? We saw that our method is reliable, first of all, because we have a pretty good assignment rate, low error rate, less than 1% error. ADMA appears more common than SDMA. There's appears to be just more of it, which has been shown. And the big thing is that most of the dimethyl peptides don't show a neutral loss. That's this huge section here where we don't know if it's asymmetric or symmetric. So that's what we want to get. We want to get more ADMA versus SDMA discrimination. So we looked back at our synthetic data because it was fragmented at multiple energies when it was run. And when we separated the results by fragmentation energy, we were surprised to see that the neutral loss did increase by a substantial amount as we ramped up to about 35 collision energy in the HCD chamber. We also saw that as we ramped up the energy, the number of neutral loss ions per spectra was increasing. So we were just getting more and more neutral loss ions. This is helpful for localizing the dimethyl site, as well as if there's multiple dimethyls in a single peptide, we can see if they're all asymmetric or if one's symmetric and one's asymmetric. So it's a very powerful tool to increase the neutral loss ions. So this is all great, but how about some real peptides? Does the same effect carry over? And so we developed a workflow to test this, where we took one fraction of our strong cation exchange and we ran it at various fragmentation energies up to 42. And this is what we saw. We got really interesting results. We saw a huge increase in asymmetric annotation, a modest increase in symmetric, but that's expected because there's less of it. But another nice surprise was that we got more dimethyl peptides just by fragmenting at a higher energy. As you can see, the bar graph got taller, which was great because dimethyl peptides can be hard to detect sometimes. So now that we saw that the collision energy works for real peptides, we wanted to do an optimized workflow where we run all of our SCX fractions and an asymmetric dimethyl arginine immunoaffinity purification with antibody. And we use 26 as our normal collision energy and 32 as our optimized. Most proteomic experiments are run at 26. And when we did this in the real data, we were happy to find that in both enrichment techniques, the increase in neutral loss was conserved for both ADMA and SDMA, as well as the increase in total number of dimethyl peptides. So everything that we saw in the synthetic data carried over to the real data. And in the end, we were able to increase the annotation for asymmetric and symmetric arginine by more than double, by 125%. And we increased the dimethyl peptide identification rate by 17%, which was pretty good. And so in conclusion, we demonstrated that higher normalized collision energy in the mass spectrometer allowed for more neutral loss assignment between these two isobaric modifications. We improved the dimethyl peptide identification rate, which is nice because these methyl peptides are hard to measure. And we developed a strong tool set to interrogate the methylome of these proteins and see how they're changing with response to knockouts or upregulation or drug treatment or inhibitors. So we're very excited to keep developing this workflow. And with that, I'd like to thank my PI, Dr. Graham, who's a wonderful mentor, taught me everything about mass spectrometry, as well as all the PhD students in our lab and the undergrad, especially Chris Liu, who helped directly with this project, and our collaborators who have also helped us in the past. With that, I'd like to thank you, and I'll take any questions. Thank you so much.